Assalamualaikum. Today I am going to discuss about the structures of protein. This is a very much important topic both for SAQ and Viva examination. So, here it is. There are four different structures. Protein have four different structures, that are primary structure, secondary structure, tertiary structure and quaternary structure. Primary structure is the linear sequence of amino acid. Here amino acid just bind together by peptide bond. That means in primary structure only one bond is present, that is peptide bond. It is the simplest structural conformation of protein. Then secondary structure. When primary structure more folded, it forms secondary structure. Secondary structure may be in helical conformation or in pleated sheet like conformation and produced by periodic coiling twisting of primary structure. That means primary structure twisted to form secondary structure. So, it is usually found in fibrous protein, and to stabilize this structure, to stabilize this folding, to stabilize this conformation, here hydrogen bond present. That means in secondary structure there are two types of bond present, that is peptide bond, without which protein is not protein, and hydrogen bond to stabilize the secondary structure, and also the secondary structure is a two-dimensional structure. For example, collagen, this is a protein which usually found in secondary structural form in our body. Then tertiary structure, when the secondary structure more folded, it is formed tertiary structure. So, it is produced by further folding and twisting of secondary structure. It is a three-dimensional structure. That means the secondary structure further folded or further tangled to form the tertiary structure. In tertiary, as it is the more folding form, or it is the more molded form, or it is the more tangled form. So, there are two sides, hydrophobic sides and hydrophilic sides. The hydrophobic side usually present interiorly, that means inside, and hydrophilic side usually present exteriorly. And this tertiary structure, this folding structure, this three-dimensional structure is stabilized by hydrogen bond, ionic bond, van der Waals bond. These types of bond usually needed to stabilize this tertiary structure. For example, albumin, globulin and antibody usually present in our body in tertiary structural conformation. We have to remember that protein are only functional in their native structural form. That means, if any kind of destruction occurs in their structure, that is secondary structure, if secondary structure destroys or tertiary structure destroys, protein will have no function in our body or cannot perform their functions in our body. Then the final structural form is the quaternary structure. It is the multi-chain oligomeric protein. That means, in this structural conformation there are multiple polypeptide bonds. In case of primary, secondary or tertiary structure, there is only one polypeptide chain usually present, but in case of quaternary structure, it is a multi-chain oligomeric protein. That means, here multiple polypeptide chain usually present. That is why it is known as oligomeric. Multiple means two or more polypeptide chain. So, polypeptide chain shows spatial relationship among each other. That means, they have own space to form this structural conformation, and individual polypeptide chain known as subunit or monomer. That means, the individual polypeptide chain is the quaternary structure. Each polypeptide chain in the quaternary structure is known as subunit or monomer form of this quaternary structure. To stabilize this quaternary structure, to hold together this multiple polypeptide chain, there should be some bond present other than the peptide bond. So, these bonds are hydrogen bond, ionic interaction and hydrophobic bond. These bonds are responsible to hold together this multiple polypeptide chain in quaternary structure. For example, there are different types of quaternary structure. Dimer means here 2 polypeptide chain present. For example, creatine phosphokinase is a good example of dimer. That means in creatine phosphokinase there are 2 polypeptide chain present. Trimer means here 3 polypeptide chain present. For example, G protein is a trimer. Tetramer here 4 polypeptide chain present, and hemoglobin is a very good example of tetramer. Pentamer here 5 polypeptide chain present, and immunoglobulin is the example. Immunoglobulin M actually is the good example of pentamer, and hexamer here 6 polypeptide chain present. So, that is all for today. Thank you so much.